Hello, good evening, everyone.
Good evening.
Uh, so, what we are going to do today, we will talk about the polymerase chain reaction and its variants. We will talk about. There are three number of variants are there, but we will talk three or four important variants and other variants introductions. We will see it. So we are going to start with the polymerase chain reaction and its basics because most of you might be knowing about the PCR, polymerase chain reactions, what is the PCR and how it works. So if you'll see, first we'll see the basics. That is a polymerase chain reaction. That is a techniques normally to amplify a small fragment of DNA. Whenever we are going to start a reactions, if from any site, any criminal places, or from any sample, if we have received a very a small DNA fragment, and that DNA fragment, if you want to amplify in larger quantity, for that one we are going to use this PCR. So the purpose of PCR is to increase the number of DNA, amplify the quantity of DNA what we are having. So this PCR was it is not a very means old techniques. It has started in 1984-85 by an American biochemist. His name was Carey Mullis. He was working at Centus Corporation, and he was awarded for this prize, this work by Nobel Prize in 1993. You might be surprised that even a single laboratory, biochemical laboratory, molecular biology laboratory, you cannot imagine that it going to work without this PCR. Means every laboratory, today's laboratory, all the molecular biology labs certainly have PCR. Without PCR, they cannot perform, they cannot go even a single experiment. If you see this using PCR is a copy of very small amount of DNA sequences are exponentially amplified. Like if you are having one copy of DNA in one reaction, you can make it two. In the second reaction, you can make it four. So it is increasing exponentially, means one to two, two to four, four to eight, eight to 16, like that one. And it is a cyclic process. We will see later on how cyclic process occurs and what are the different components we require it. So PCR is now a common and most indispensable technique. Means you cannot replace PCR with any other technique, any other instruments. So that's how it's called as indispensable techniques used in medical laboratory, diagnostic laboratory, research laboratory, whatever labs you can talk about, whatever labs you will think about, any kind of labs, any kind of works where DNA sample we are going to use it, there certainly we require PCR. Most of the PCR methods based on the thermal cycling process. What we are going to call thermal cycling process, because thermal means temperature. So during the process of this PCR, we are going to increase the temperature, we are going to decrease the temperature. So in a cyclic manner, temperature go up and come down. Okay, so that's why it's called a cycling process and thermal cycling, because temperature ups and downs we are doing in a cyclic process. And the instrument which where we are using this thermal cycler process in this PCR, that is called as thermal cycler. So the instrument is called as thermal cycler, and normally process is called as thermal cycling process. If where what is the PCR used for, where we are going to use it, I have told you, in any biomolecular biology labs, in any field you can imagine, and every field we are going to use this one. So it may be environmental microbiology, medicines, phylogenetics, bioinformatics, forensic science, food technology, agricultural fields, or some type of genomics. So everywhere wherever DNA sample we are going to use it, there PCR will be applicable. So what we are doing it, if you want to selective DNA isolation, there also we are going to use PCR. For the amplification and the quantification of DNA, there also we can use PCR. For medical application, we can use PCR, like for the genetic testing of some specific genetic disorders, like inborn error of metabolism, or any other genetic disorders, any other genetic complications, single nucleotide polymorphism, any kind of proto-oncogenes, any kind of oncogenes, tissue typing, uh, then infectious disease applications, their diagnostic, DNA fingerprinting, criminal investigations, DNA paternity testing. So many places we are going to use and then ultimately lastly result applications for the process. Nowadays, you might have also heard about that in the case of COVID. There also we have used this PCR too much, not PCR directly. It was a reverse transcripted PCR, RT-PCR, because the genetic material which virus is having, that is RNA. So that's why we have used reverse transcripted PCR. Is it correct, right?
Yes.
Fine. Wherever you will feel any kind of complication, any kind of issue, you can stop me. Fine. Now, so this is the application of where we are going to use it. If you will divide application of PCR in three broad area, we are having three broad areas are like it will be. Just give me 1 second. So we are having this one, this molecular identification, sequencing, where we are going to have genomic gene cloning, human genome projects, and other things, and genetic engineering like site-directed mutagenesis, SNP analysis, mutation analysis, proto-oncogenes or oncogenes identifications, and gene expression, where we want to see any kind of gene expression. So all these applications are PCR. So I have told you, you cannot think of a lab of molecular biology where PCR is not going to be used. So PCR is applicable in almost all the labs, all the molecular biology labs. Now, what is the basic steps of PCR? If you see, there are three basic steps of PCR. So where we are going to start with one basic double-stranded DNA. This double-stranded DNA is first, in the first step, we are having a denaturation step, where we are going to denature the DNA by heating DNA. You know that DNA is joined with the hydrogen bond. So two strands of DNA have hydrogen bonds. These hydrogen bonds are very weak bonds. These are non-covalent bonds. So whenever you are going to heat DNA, this hydrogen bond will be broken. And as soon as this hydrogen bond will be broken, DNA will be separated out. So both the strands has been separated out. This is called as denaturation step. So normally this process, this step takes place at very high temperature, that is roughly 92 or 95 degrees centigrade. Whatever DNA you have separated in this denaturation step, that DNA is going for second step, that is called as annealing step. Annealing means joining step. We are going to have a primer. Primer we are going to call a small DNA fragment which is roughly 10 to 20 base pair nucleotide nitrogenous base pair long, and this is complementary to your target DNA. So we are going to have a reverse primer and the forward backward primer, reverse primer, or the you can call it forward primer. These primers are going to, in the second step, we are going to decrease the temperature from 94 to 50 to 56 degrees centigrade. This annealing temperature varying. So according to the primer, later on we will talk about that one. So this denaturation step, after denaturation step, we are going to reduce the temperature to 50 to 56 degrees centigrade, so that their hydrogen bond will form between this primer and your DNA. Because if you will not decrease the temperature, hydrogen bond will be not formed. So we have to decrease the temperature so that hydrogen bond will form at this temperature. Clear? So this is annealing temperature, and the third and last step is the extension step, where DNA polymerase will be there, and DNA polymerase will elongate the primer according to the DNA. So this will work as a template, and complementary strands will form here. So here also and here also. This is called as elongation step or extension step, where DNA polymerase will do. So after annealing step, we have to rise the temperature, increase the temperature up to 72 degrees centigrade. So we have started with 94, came back to 50 to 54 degrees centigrade, and then again reaches to 72 degrees centigrade. So you are going to see the temperature is going up and down in a cyclic manner. So that's why it is called as thermal cycling processes. And remember, three stages: denaturation stage, annealing stage, and extension stage. Any confusion, anything? Is it clear? Fine. So this is the simple process. We have started with single DNA, and as a result, we have got two DNA. So we have started with one DNA fragment, and as a result of this three step, we have got one DNA and second DNA. So this is, you can call it one kind of cloning process, one kind of replication process. But this is a cyclic process. By single step, you can get only two fragment, two pieces of DNA. So how it is working in the cycling process? Let's see, this is, the process will be the same. We are starting with one single DNA. In the first stage, it is going to be denatured. So two separate DNA have been strands has been separated by fragmenting by heating process. So hydrogen bond will be broken and two strands has been separated. After denaturation has done at 90 to 95 degrees centigrade, then annealing process will takes place where primer will join with the forward and reverse strands. So this is annealing process, second stage. In the third stage, this elongation will be there. So DNA polymerase will elongate this strands here and this strands here. So as a result, we have got two pieces, one piece here, one piece fragment of DNA here. In this second, this is called as one cycle. So process number one, process number two, and process number three. These three processes cumulatively called as one step of the one cycle of the PCR. After this one first cycle of the PCR, this two DNA fragment will again go for denaturation step. So this will denature and give you one fragment and two fragments. This will also denature, give you two different fragments. So as a result, when it will start the second cycle, you will have four single stranded DNA. So ultimately, this cycle is going on. Now, when you are going to start the DNA, this was the basic process, but there are certain requirements are also there. What are the requirements we are having for PCR? The first requirement is you require a DNA template. What DNA template? The target which you want to elongate, which you want to copy, which you want to increase. So that will be your DNA template. Then we require the primer strands, which will be a small DNA fragment complementary to your target DNA, both forward and reverse strands. So primer which is complementary to your target DNA. You might have heard about primer during your DNA replication process. Whenever you have studied your DNA replication, that time you might have heard about this one. So we require DNA template, we require primer. For the elongation, we require a DNA polymerase. And you have seen that because normally all the DNA polymerase which we are having, they works at normal temperature because our body temperature is 37 degrees centigrade. You have seen that we are going to increase temperature up to 92 degrees or 72 degrees. Certainly normal DNA polymerase will not work here. So we require a thermostable DNA polymerase which will not be denatured at 92 degrees because proteins are going to be denatured. If you are going to increase the temperature, protein will denature, and you know that all the enzymes are proteins in the nature. These proteins, if you will heat at 92 degrees centigrade, proteins will be denatured. We don't want that one. So you require a DNA polymerase which will be stable at 92 degrees centigrade or 72 degrees centigrade. So that's why we require a thermostable DNA polymerase. I will talk in detail about that one. Then we require for replication, because you have to add a nucleotide, your enzyme has to add a nucleotide here. So they require adenine, thymine, cytosine, guanine, so that whenever cells require here adenine, they will add here. Whenever they will require thymine, they will add it here. So they require all the four deoxynucleotides. Remember, it is deoxynucleotides. Okay. It is deoxynucleotides. Now, fourth thing is we require buffer. Buffer means a kind of environment which provides the system to the enzyme so that it will do the process. Clear? So these are the basic requirements. So in buffer we are going to provide, or we are going to provide some kind of cofactors which help our system, which help our body means tube, so that it will do the actual process of the elongation or the polymerization. So this is the basic requirement we are having. Now, if you see the in the details, what are the requirements we are having? So we require a DNA target DNA template. Remember, you either, if you want to take a snapshot, you can take a snapshot, or later on I will give you the slides also. So we require target DNA template. We require thermostable DNA polymerase. Thermostable means those enzymes which will be stable at higher temperature, not denatured. Then we require two DNA primers, which is complementary to the 3' end of each of the sense and antisense. You know that what is sense and what is antisense. I have also talked many of times. That is what is template, what is anti-template, what is sense, what is antisense. So both extents, one extent will be sense, other extent will be antisense extent. Then we have to provide deoxyribonucleotides, all the nucleotides in the deoxy form, because it is DNA replication. Then we require buffer solutions which will maintain the pH and other things. I'll repeat it. In our DNA, I will finish this one, one things, and then I will talk about bivalent cations. We most commonly, whenever we are doing performing PCR, we require bivalent cation ions, means two positively charged metal ions. Either it will be magnesium or manganese most commonly. But if we don't have this one, then you can use potassium also. You can use monovalent cation also. But we require these things. These are the cofactors. Okay. What is called as sense and antisense? See, we are having normally double, our DNA is double extended. In both the extents, we are not going to use both the extents for protein synthesis. So whenever this DNA is going for transcription process, only one extent will be used for transcription process. So one extent we are using, and we are making a complementary to the that particular extent. Like for example, I am going to use this extent as a for my transcription process. So this extent I will have here, and I will make a complementary RNA molecules, RNA component. Because this extent I have used from the DNA, I have make a complementary of this one. So this extent is complementary to this one, and this RNA is also complementary to this one. Means this extent is common. So this and this, the DNA which we have not used and RNA which we have formed, are almost exactly same in the sequence, except thymine in the DNA and uracil in the RNA. Correct? So the strands which you are going to use for transcription process is called as antisense strands. This is called as antisense strands, and the strands which you have not used, that is called as sense strands. Why we are going to call sense strand is because the sequence of the sense strands is similar to the RNA. So we are going to use sense strands, or the sequence of the sense strands will be for protein synthesis. So this sequence is similar to this sequence, which DNA you have not used it, and that is called as sense strands. So this is sense and antisense. So remember, for transcription process, we are going to use only one strand for transcription, that is antisense strands, making a complementary RNA that is similar to sense strands. Clear? Fine. Now, what is thermostable DNA polymerase? Fine. What is thermostable DNA polymerase? If you will say, there are four different, mute yourself, those who are, okay. So whenever we are going to use enzymes, there are four important thermostable enzymes which commonly we are going to use it. What are those enzymes? Like first and most common, yeah, most primitive. The first enzyme which was discovered for this PCR was Taq polymerase, which was discovered from Thermus aquaticus. So Taq polymerase means T from here and AQ from here, so it becomes Taq polymerase. This was discovered from U bacteria. So this is called as Taq polymerase. If you want to, you can write the names. Second one is PFU DNA polymerase, which was discovered from Pyrococcus furiosus. That was means Archaebacteria. Third one is the Vent polymerase, which is isolated from the Thermococcus littoralis, and that is also Archaebacteria. And the fourth one is TWO polymerase, that was isolated from Pyrococcus vosei, that was also archibacteria. So the first enzyme was from eubacteria and remaining all three is from archibacteria. Clear? Now, there are certain which enzyme you should use it depending on the availability, also depending on the cost also, but there are certain advantages and disadvantages which these enzymes are having. Like the thermotac polymerase is having advantages that this enzyme is the most primitive enzyme, most first discovered enzyme for polymerase chain reactions. So that was the its significance, and its optimum activity occurs at 75 to 80 degree. So that's why if you see the elongation step occurs at 72 degree centigrade. The third step, first 92 to 94, second 50 to 54, and third elongation was 72 degree centigrade. Fine. So the optimum temperature of Taq polymerase is at 75 to 80 degrees centigrade. The half life, as you are going to heat them, the if you will heat the enzymes, normally their activity is going to decrease or going to increase. Fine. Now, the half life of this Taq polymerase is 2 hours if you are working at 92 to 95 3 degrees centigrade. Half life will be 40 minutes if you will heat it at 95 degrees centigrade, and it will be 9 minutes if you will heat more almost 100 degree 97.5 degrees centigrade. So the thermostability of this enzyme is very high. This Taq polymerase can replicate 100 base pair per second, means 1000 base pair in every 10 minutes, sorry, 10 seconds. So you can remember. And commonly we are using temperature at 72. So at 72, it can add, this enzyme can add 1000 base pair per 10 seconds. Now, one of the drawback of this Taq polymerase is they don't have any proofreading activity. Normally, proofreading activity means 3 to 5 exonuclease activity. So, whenever wrong nucleotide will be added, they cannot correct it. So, if you are using Taq polymerase for your processes, for your what we call it PCR reactions, you may have some kind of mutations in your DNA. If some wrong nucleotide will be added, Taq polymerase cannot correct it. So that is the major drawback of this Taq polymerase. Best enzyme which we are having, that is the PFU polymerase, which is having the better thermostability than the Taq polymerase or any other enzyme, and also they are having proofreading capacity. So you can write it if you are going to get a question that which thermostable DNA polymerase is the best and why. You have to write PFU polymerase is the best because it is more stable at the higher temperature and it is having the better proofreading compared to the Taq polymerase. Unlike Taq polymerase, PFU DNA polymerase processes 3 to 5 exonuclease, means it is having the proofreading also, so it can correct your wrong nucleotide addition also. So you will get a corrected nucleotide, corrected DNA, no other nucleotide. Third component which we needed is the DNA primer. This primer we require two sets, one for your sense strands, other one is for your anti-sense strands. Now, generally, most commonly we are using 18 to 20 base pair nucleotide long primer. Your primer should not be too longer and it should not be a smaller, very smaller also. If you will use too long primer, it will not bind in your DNA. And if your primer will be very small, it will bind at multiple places. So, you can call it non-specific. If your primer length will be very small, non-specific binding will be there. Wherever don't want it is not required, there also it will bind. So that's why primer length should be very specific. It should be 18 to 20 base pair long. And chemically synthetic primers are available in the market. If you give the DNA strands, DNA sequence to any company, they will provide you artificially synthesized primer will be given. Online also, if you put the sequence online in the different tools, we are having online on NCBI, we are having primer designing tools. Many software we are having for primer designing tools. So if you will give your sequence, they will give you the primer design, primer sequence. They will give it to you. That sequence you will send it to the company, and company will provide you the primer oligonucleotide. This complementary primers is going to add at the 3' end of the target DNA on both the strands, primer forward also and backward also. This primer should not have any internal complementary regions. Like if you, this is your primer strands, these things, this strand, this portion of the strand should not be complementary to this one. If this will happen, then what will happen? Your primer will bind with each other and it will form a loop-like structure like this one. So if complementary regions will be there, they will join together, it will form a complementary. So remember, when you are going to design a primer, no complementary region should be there, neither within the primer also, nor both the primers should have complementary region. So these are the different kind of conditions you can call it for primer designing. Should not contain any inverted repeats. If you are having inverted repeats, then this situation will occur. Hairpin loop will form. That we don't want. In your primer, if you are having more GC content, for roughly on an average, we require 40 to 60% guanine to cytosine content for better annealing, because a strong binding will be there. GC will have three hydrogen bonds. If more AC content, AT content will be there, only two hydrogen bonds will be there. So as much as GC content will be there, that much better annealing will be there. So roughly 40 to 60% GC content will be there, because in one step primer is going to bind to your DNA, and during second cycle it has to be denatured also. So that is called as melting temperature. TM means melting temperature of primer can be calculated to determine the annealing temperature. Annealing means binding, and melting means when it will be denatured. So the melting temperature and annealing temperature both are important. These are some of the formula uh, for the melting temperature calculations. You don't require, when you are going to use software tools for primer designing, you don't need this formula to be remember, because software will detect it. Otherwise, you can, if you want, you can remember it, this one also. Now, we are going to see the three steps in details, and then we will go further. This step is first step is we have seen that first step was the denaturation step, where we are going to denature the original DNA, which is a double stranded DNA. So in the first step we are having two double stranded DNA, where which will bind with the hydrogen bonds. So in this case, we are going to heat the reaction chamber to 94 to 98 degrees centigrade, roughly 201 to 208 Fahrenheit for 20 to 30 seconds. And if you are going to heat the DNA, so what will happen at this temperature? Hydrogen bond will be broken. When hydrogen bond will be broken, DNA strands will be separated out. So as a result of this first denaturation step, you will get two separate DNA, single stranded DNA fragment. Roughly the time duration required for this process is 30 to 90 seconds. So this is the original DNA, which is the unamplified target DNA, which is having the two strands. After denaturation step, you can see the hydrogen bonds has been broken and both the strands has been separated. So this is one strand, this is second strand. Now this both separated strands will go for the second step, that is the annealing step. In this annealing step, initially we have increased the temperature to 94 degrees centigrade, which we have seen here. The temperature has been increased to 94 to 98 degrees centigrade. Now, what we are going to do, we are decreasing the temperature to 50 to 65 degrees centigrade, depending on the primer length. Again, if more primer will have higher GC content or lower GC content, length of the primer, these temperature is going to vary. That's why we have a range. We don't have a fixed temperature. So we are going to decrease the temperature from 94 to 50 to 65 degrees centigrade for again 20 to 40 seconds. And when you are going to decrease the temperature, whatever hydrogen bond was broken in the previous step, that hydrogen bond between the primer will form. So this step is very critical, because if annealing will not there, if primer will not bind there, then elongation will not occur. Now, what is the problem going to happen? See, you are having DNA. See, this was the two DNA has been separated, and you are having primers in between. When you are going to decrease the temperature at 50 to 60 degrees centigrade, this DNA is complementary to this DNA. So it might be possible that these two DNA will form, join together, form a hydrogen bond. If this is going to happen, this is of no use, because it will not go for your PCR reaction. If any primer, this primer will join with this one, form a hydrogen bond with this one, that will of no use. If in primer will form internal hydrogen bond, that will be of no use. So exactly what we require it, the primer should bind at this particular place here, and primer should bind at this particular place with this one, so that this is called as actual annealing. So annealing process is one of the very important step in the PCR reactions, where primer will bind with the DNA, target DNA, not two DNA, original DNA will bind, join together, no two primer will join together, no internal repeats will be there. So exactly this is the critical step in the process. Now, the temperature should be low enough so that hybridization will occur. Means you are decreasing temperature to 50 to 60 degrees centigrade, so that hydrogen bond should form. If your temperature will be very high, it will not bind. If your temperature will be very low, unnecessary binding will be there. So temperature you have to judge very carefully. Software will give you this temperature also depending on your primer sequence and the primary thing. A rule you can remember that this is a rule where annealing temperature means the temperature at which this primer is going to bind with the DNA. So the annealing temperature is roughly 3 to 5 degree low than melting temperature of the primer. Remember, if melting temperature of a primer is 55 degrees centigrade, so annealing temperature will be around 52 degrees centigrade, roughly 3 to 5 degree less than the melting temperature of primer. Write down in your notebook. So whatever DNA you have separated in the first step, so this is one stance, this is the second stance, and these small blue and green fragments you are going to see, these are the primers, which he has written here. So these primers, when you are going to decrease the temperature, this should be 55, not 45, this should be 55. So when you are going to decrease the temperature to 55 to 60 or 65 degrees centigrade, primers are going to bind with the DNA. So as you can see here, this is the actual binding. Primer will bind with your target DNA. This is called as annealing process. Annealing means joining of primer with the target DNA. If blue and green primer will join with each other, this is of no use. If this DNA will join with this one, this is of no use. But these things happens, but it's of no use for myself. For me, only these two sequences are important because this will go for primer PCR. Here is a question for you, MCQ. Uh, why Vent and PFU polymerase are efficient than the TAC polymerase? And you can write in the chat box. Proofreading was the one thing I have told you, and second thing I have told you, higher thermostability they are having. So this can be in the means you can get in the, you have to write also. So there you have to write details. Perfect. Fine. Now the third step is extension step or elongation step. In this case, the temperature depends on the DNA polymerase. If you, because DNA polymerase you have seen that we are getting from the bacteria. So different enzyme, different bacteria lives at different temperature. So from wherever this you are going to get this DNA polymerase, depending on that one, your enzymes temperature depends. The optimum temperature is roughly between 75 to 80 degrees centigrade, or on an average you can call it 72 degrees commonly used for any kind of DNA polymerase. So there we are having 72-degree optimum, and the time duration required is 45 seconds. So if you see, it is almost less than 1 minute for one cycle. First cycle 20 to 30 seconds, second step 20 to 30 seconds, third step 45 seconds. On an average, it is 1.1 to 1.5 minutes for one cycle. The precise time required for elongation depends on two basic characteristic features. One, how which DNA polymerase you are going to use it, because every DNA polymerase is having their own strength. One DNA polymerase elongates very fast, other DNA polymerase works very slowly. So depending on the strength of DNA polymerase, and second, how longer DNA you are going to target. So the length of the target DNA also decides how much time it will take, and the power of the DNA polymerase also decides. Roughly, on an average, every DNA polymerases 1,000 base pair per minute. Okay. So this is the third step where primer, you can see these fragments which you are going to see. These are the nucleotides. These are the nucleotides. And primer has been annealed in the second stage. And third stage, DNA NTPs are there, and we are having DNA polymerase, thermostable DNA polymerase. These are going to elongate. DNA polymerase will elongate, add a nucleotide here also and here also. So as a result, you are getting two different DNA. We have started with one DNA piece, and as a result, we have got two DNA fragments. So this is the one cycle of the PCR. You can see we have started with one DNA. After denaturation, annealing, and we have got two DNA. So this is called as one cycle of the process. In the second cycle, you will get these two DNA will be used, and you will get ultimately four DNA. So at the end of second cycle, we have got four DNA. So this is called as exponential growth of the DNA. So this is overall all the three stages. We require a DNA. This DNA is going to be denatured at 95 degrees centigrade. So you will have two separate DNA strands. In the second stage, we are going to add DNA polymerase, nucleotides, different DNA primers, and annealing will be done at 50 to 55 degrees centigrade. So primer will be added here on both the strands. In the third stage, DNA polymerase will elongate this one, both the strands, and as a result, you have got two DNA strands. So this is overall one cycle of the reaction, one cycle of the PCR. Roughly, for a sufficient quantity of DNA, we need to do this process 20 to 40 times. Yeah, 20 to 40 cycles need to be repeated. Then only you will get sufficient quantity of DNA. This is the second question. At what temperature denaturation of DNA double helix takes place? What is the optimum temperature for DNA denaturation in the PCR reaction? 94 to 95. Perfect. Fine. Now, if you'll see the temperature, this is the complete PCR cycle. We are starting DNA at the denaturation step where temperature will be 94 degrees centigrade. So DNA has been denatured here when you are going to heat temperature at 94. Then you are decreasing the temperature to 50 to 55 to 65 degrees centigrade. So here annealing will take place. This is annealing step. Once annealing will be done for 45 seconds, then you are again going to increase the temperature to 72 degrees centigrade. So this is from here to here is called as one cycle. Denaturation at 94, annealing at 54, and extension at 72. This cycle we are going to repeat multiple times, 20 times, 30 times, 40 times. And once this process will be finished, uh, your PCR instrument, your thermal cycler will keep your DNA at 4 degrees centigrade. Whenever you will come back, you will get your DNA sample. So this is the complete PCR process. There are some points which we need to remember is the process of denaturation, annealing, and elongation makes one cycle. As you have seen here, one denaturation, one annealing, and one extension is making one cycle. Multiple cycles are required to amplify the DNA target to millions of the copy. The formula which by which, like if someone is going to ask you how many copies of DNA you will get after 13 cycles, after 10 cycle, after 15 cycle. For that one, we are having a formula that 2 to the power n. N means n number of cycles. Like if we are going to run PCR for 30 cycles, so how many DNA you will get it? 2 to the power 30. If you will solve it, you will get this value. Means if you will start with one DNA and run 30 times PCR cycle, 30 PCR cycle, this many pieces of DNA, this many copies of DNA, you will get it. So this is exponential growth. Okay. Okay. This questions will come later on. Now, you see, this is one DNA, and separation has been done. Then here annealing has been done, and extension has been done. So this is one cycle. One DNA give you two copies. In the second cycle, we have got four copies. Third cycle, you have got eight copies. 30 cycle, you will get 10 power 94 9 size. Now, one important thing in this case is, if you will see after second cycle onwards, this was the original DNA. This was the length of my original DNA. But if you will see after second cycle onwards, means from the third cycle, like this DNA, if you compare with the original DNA, the length of this DNA is smaller than this one. Why it is smaller? Because your primer, if you are going to, this is the primer which is not binding here. If primer will bind here, then elongation will be up to here. In this case, primer does not bind at the terminal place. Your target DNA may be somewhere inside. So primer binds here and elongation will occur after here. So this portion of DNA has been left off. Like, see here. If primer binds here, so elongation will done for this part. So this portion of DNA has been left out. So in after the second cycle onwards, from the third cycle onwards, you will get some pieces of DNA which does not have this fragment. Similarly, this side also, this DNA fragment has been lost. Like see here, in this copy of DNA, in this newly formed DNA, you don't have this copy, this portion. In this newly formed DNA, you don't have this portion. When this DNA will be elongated later on, you don't have, you will have a similar DNA. So question is, why after second cycle onwards, yeah, from third cycle onwards, DNA length getting smaller during the PCR? The reason is because primer does not bind at the terminal portion of the DNA, whatever DNA fragment we are going to use it. If primer binds at the terminus, then complete DNA will be elongated. But if by target will be primer bind somewhere inside, the DNA will be elongated only for further. Whatever portion has been left on the primer during primer binding, that portion will not be elongated. Is it clear for everyone? Fine. So this is one of the important question reasoning, that why primer length is getting smaller after third cycle onwards or after second cycle. Now, the primer efficiency, how good PCR you are doing, how good PCR product you will get it, how good PCR result you will get it. There are certain processes, certain components, certain things like efficiency of amplification, yeah, PCR decreases as the length of target DNA will be longer. If your DNA is very long, the efficiency will decrease. Efficiency of your primer is also affected by the primer length. I have told you primer length should not be very long, should not be very small. So the primer length is also going to affect the efficiency of the PCR. Then sequence of the primer means GC content. Yeah, go on, internal sequences, internal repeats are there, complementary are there or not. So GC content and internal repeats, that is called as primer sequence. Then annealing temperature, binding temperature, guanine cytosine content. Sometimes some chemicals you are going to use, some reagents you have used it which inhibits your enzymes, which inhibits your PCR. Some of the PCR inhibitors are phenolics, humic acid, blood degradation product. Like if you are going to get DNA sample from the blood, and that time heme will be left out in your DNA sample. So that heme is going to inhibit your PCR. So phenolics, humic acid, heme or heme breakdown product, you can call it bilirubins or some other products. They normally decreases your efficiency of the PCR. Now, commonly what we are going to do, we are going to use bovine serum albumin in the during the PCR. Normally, the purpose of using BCA or bovine serum albumin during the PCR is it protects your DNA polymerase from these inhibitors. So even if you are having some kind of inhibitors in your PCR process, those will be protected by using this BSA, bovine serum albumin. So this was PCR. If you have any question in the PCR, you can ask anyone. Any question you have? Okay, fine. So now we are going to see the variants of the PCR. There are a number of variants of the PCR. So we are going to start with multiplex PCR. As its name suggests, you might have gone through the watching movie in multiplexes. What is the meaning of literal meaning of multiplex? Can anyone will tell me? What is the literal meaning of multiplex? नहीं नहीं बता पाएंगे. Multi means many. We are going to watching the movie multiplex means many. You can call it the theaters hall will be there. Many halls will be there. Uh, so that is called as multiplex. Normally in the PCR, what we have done it, we have done only one primer. We have used it. Only one kind of one pair of PCR we have used it. In multiplex, we are using many, more than one. So that is called as literal meaning of multiplex. So this process normally amplify DNA sample using multiple primers. We are not going to use only one pair of primers. We are using multiple pairs of the primer. Okay. So that is called as multiplex primer. That is the literal meaning. Now, and it is temperature mediated PCR reactions by the same thermal cycle. Every process will be same. Only thing is we are going to use multiple primers. We will use it. Multiplex PCR was first described in 1988 as a method to detect deletion of the dystrophin gene. You might have heard about dystrophin gene. This is a gene which is one of the probably one of the largest gene we are having, largest protein it gives you. Go and search what is this significance of dystrophin gene. This is one of the largest gene we are having. So the multiplex PCR was first described in 1988. After the PCR has been discovered, PCR was discovered in 1985, and this multiplex PCR was discovered in 1988 as a method to detect deletions, any nucleotide deletions or mutations in the dystrophin gene. In 2008, this multiplex PCR was used for the analysis of microsatellite genes or single nucleotide polymorphism (SNPs). In 2020, this RT-PCR multiplex assay were designed which combined multiple gene targets from the center CDC in a single reaction and which was used for the detection of COVID-19. The primer designing for all primer pair has to be optimized because you know that every pair of primer have their own melting temperature, own annealing temperature. If you are using four different pairs of primer, so four different type of temperature you will have it. So which temperature we should use it? So that you have to judge it. Normally we are going to select such type of primers whose temperature would be similar or almost in the same range. If you should not use two different primers or more than two primers whose temperature, annealing temperature will be very different. Like one works at 70 degree, other one works at 78 degree. You don't should not use that much. So we are going to select such primers which will work at almost similar annealing temperature. Multiplex PCR is the simultaneous detection of multiple targets in a single reaction well with a different pair of primers for each target. Like once you will see the example picture, you will understand it. Like, see, in like this is the traditional PCR. Like if I want to detect four different DNA, I will use one primer set here, one pair of primer here, one pair of primer here, one pair of primer here. So I will have to run four different set of reaction. One PCR for this one PCR for this one PCR for this one PCR for this one. So we will have four different primer set, four different reaction mixture, four different components. Up in case of multiplex PCR, what we are going to do it? All the four samples or the four primers we will put in the single tube, and the single tube will run PCR and then later on we will separate them. That is called as multiplex PCR. What is the advantages? One major advantage is it you are not going to require enzyme in four different tubes. If you will do traditional PCR for four different samples for four different primer, you require enzyme here also, here also, here also, here also. In case of this one, you require only one places enzyme, so you are going to save the resources. That is one of the major advantages of this one. Okay. So this multiplex PCR is used in life sciences research, clinical diagnostic, and forensic sciences. Mainly, we are going to use for diagnosis of important foodborne diseases. So, we are going to run PCR. We are having one DNA, second DNA, third DNA, fourth DNA. We are going to use four different sets of primer. All the primer will be put in the single tubes. Run the PCR and then separate them because every DNA will have their own length. So once you are going to separate the DNA along with the electrophoresis, four DNA according to the length you will separate it. So you will get four different separate elongated DNA. What we require it, again similar things we require it, only difference is in this case this primers we are going to have multiple primer set. So we are having template DNA, either you will have only one template DNA or you can have multiple template DNA from multiple sources, and accordingly you can have multiple pair of forward and reverse primers. If you are using four different DNA, you want to use four different sets of primer. Nucleotide polymerase, not only tac, you can use tac also, vent also, pfu also, pwo also. So any kind of enzyme you can use it. And then you are using going to use buffer and mgcl2, or you can use it magnesium chloride, or you can use it potassium chloride depending on availability. And the same PCR thermal cycler you are going to use it. So this is the process how we are going to do multiplex PCR. What are the different types of multiplex PCR? We are having two basic type of multiplex PCR we are having. One is single template PCR reaction. In this case, we are going to have only one DNA, target DNA, and from this one we are going to use multiple primer, one for this place, one for this place, one for this place, one for this place, maybe, or more than four places. So when you are going to have the single DNA, from the single piece of DNA, you are going to detect multiple fragments. This is called as single D template PCR reaction. In this case, you will have single template DNA. Several pairs of forward and reverse primer will detect different fragment of this single DNA. Second one is multiple template PCR reaction. In this case, we can have multiple many DNA. As example, you will see it like here. This is example of uh, multiplex PCR because we are having different fragment, different DNA. So you can have one DNA from one organism, second DNA from second organism, third DNA from third organism, fourth DNA from fourth organism. So in this case, we are going to have, this is called as multiple template PCR reactions. Presence of multiple primer may lead to cross hybridization also. They will, each primer can bind with a different other fragments. So these are some of the drawbacks. Now, criteria for the successful multiplex PCR is the primer designing for all the primer pair has to be optimized so that they will have the similar annealing temperature for the second stage of the PCR where it will bind with the DNA. So this annealing temperature you need to finalize in such a way so that for all the primer pair temperature will be almost comparable, almost similar or almost equal. Primer design should ensure all primer pair will be very high specific so that no cross hybridization will be there, no cross binding will be there. Size of amplicon, size of your tube where PCR is going to take place. This amplicon size should not differ too much, and one target might get amplified while other might not. Setting the polymerization time optimally is important. For the multiplex PCR, like in case of normal PCR, we have seen. I'll talk about PCR efficiency. I will talk about. PCR efficiency, what I have told you, it depends on different parameters. It depends on how big DNA you are going to elongate, which type of enzyme you are going to use it. If your DNA will be too long, your efficiency will reduce. If you are going to use a enzyme which is not efficient, which speed will be very slow, which uh, that is not give you too much better efficiency will be there. If your primer length will be too high, that will also affect your means the PCR PCR effect, you can call it output. Then if your GC content will be very high or very low, that also going to affect annealing temperature. And lastly, there are some kind of PCR inhibitors. So normally these are going to affect your PCR efficiency. Okay, when we are going to design primer for the multiplex PCR, these are the some of the conditions. Like those things which is highlighted, they are important. As you see, know that multiplex PCR will have multiple primer sets. Usually, primer should be smaller in length, roughly 18 to 20 base pair in the length. It should be melting temperature primer with the similar melting temperature, preferably which temperature will be between 50 to 60. Not only melting temperature, its annealing temperature also should be the same. For sequence which is having a high GC content, guanine cytosine content, primer with high melting temperature are recommended. Okay, the TM variation of between 3 to 5 degree is acceptable for primer in a pool. Specificity primer should be very specific to the DNA, and normally avoid primer dimer formation, means two primers should not bind with each other. No internal dimerization, no external dimerization. Where we are going to use this multiplex PCR is going to be used. There are multiple uses of this multiplex PCR. It is going to be used for SNP genotypic, single nucleotide polymorphic genotyping, many pathogen detection. Just now you have seen that we have detected for foodborne diseases detection, genetically modified organism detection, GMO, forensic studies, food analysis, mutation and SNP analysis, gene deletion analysis, dystrophin gene analysis. Just now you have seen it. Deletion studies has been done in gene for gene deletion, template quantitations, linkage analysis, and RNA detection. These are some of the important uses of multiplex. If you will see the paper submission in the different journals, by using this multiplex PCR, it has increased tremendously. Means initially very less number of paper was submitted, but nowadays too much amount of number of PCR has been paper has been submitted to the various journals. Advantages of multiplex PCR is it is more informations with the less samples, because you require only one DNA or two DNA, or a small resources buffer enzymes also going to be used, required in very less quantity. High throughput, cost effective because only few nucleotide enzymes and other consumables will be used. Time saving, it is going to save your time. Less input material required. More data from the limited starting material is required. Increased accuracy of data normalization, and few pipetting error, because you are going to pipette in a single tube, not four times you have to do pipetting. So these are some of the advantages. Second important variance is hot start PCR. As its name suggests, it is going something related to heating. So what is hot start? The specificity when we are running the PCR, the specific DNA should be elongated. So specificity is the main aim of any PCR reaction. Every PCR modification are meant to increase specificity as well as sensitivity. Means we have a normal PCR. Normal PCR means we are having three basic steps: denaturation, annealing, and elongation. Whenever we are doing any kind of modifications, like just now you have done multiplex PCR, that is one kind of modification. We are going to see hot start PCR. This is another kind of modification. So whenever you are going to do modify the basic PCR reaction, basic PCR process, the purpose is to increase the specificity of the process, to increase the specificity and their sensitivity of the reaction. Non-specific binding is the major problem. As I have told you, primer length will be very small. It will bind at many places. Even non-specific binding will be there. So the non-specific binding increases the chances of getting false result. So this hot start PCR is a modification of the conventional PCR, which normally decreases the non-specific binding. Remember, significance of this hot start PCR is It reduces non-specific binding of primer to the target DNA by limiting one of the reagent until the heating state of the PCR. Primer, dimer, and mispaired or the false target of the major region for non-specific binding. How it is going to happen? See here. Uh, in this case, this is the conventional PCR. What we are going to do before starting PCR, we are putting everything. We are putting target DNA also, primer also, nucleotide also, buffer also, minerals also, and then we are going to put it into the thermal cycler and do the PCR. This is normal conventional PCR. In the hot start, what we are going to do? We are going to uh, stop means restrict one of the component, and one component will be added later on. Like in this case, you can say Taq DNA polymerase is associated with the cap. Here, enzyme is not going to the mixture, and so here wax will be there. Wax means this wax does not allow this enzyme to go inside. After the heating means in the first step when your DNA will be denatured. So you are going to heat at 92 degrees centigrade. That time this wax will be melt out, and after heating process this enzyme will go to the mixture. So some of the component is means all the component we are not mixing in the initial step, in the first step only. Some of the component we restrict, we limit, and that we are going to add later on. So these are the details regions why non-specific binding occur. This is not required for you. There are four important things: either because of the annealing temperature, or a specificity of the primer, or high concentration of the different components of the PCR, or early action of Taq polymerase. So any of the component, if you want to decrease this non-specific binding of primer, so you can change annealing temperature, or you can change at the primer this step, or you can change here, or you can change here. So any of the components can be restricted, so that it will does not allow the specific non-specific binding. In case of hot start PCR, one of the component starts its activity under the hot condition of PCR. Means one process, one component will do their process later on, not from the first stage. Like in this case picture you have seen, enzyme is going to start working after the heating process, after the denaturation process. The hot start PCR is the most advanced modification of the conventional PCR, in which one of the PCR reagent is activated only after heating. Like in this case, enzyme. Enzyme is going to be activated only after denaturation step. So the non-specific binding and primer dimer decreases the yield of the reaction, and our DNA interest amplifies less. So the purpose of this hot start PCR is to restrict or limit the reaction at an early stage by limiting the Taq DNA polymerase in the reactions. So you are having every component here except enzyme in this reaction mixture. So here after you are going to heat it at 94 degrees centigrade during the denaturation step, and then because of this heat wax will be melt out, and then enzymes will reach there and PCR reaction will take place. See here, everything will be there. This is the conventional PCR, and here after heating enzyme will reach here. In this case DNA normally DNA polymerase was inactive, or it was not there. Once you are going to heat it, then DNA polymerase will reach to the reaction mixture, or it will be activated, and ultimately you will have only desired product will be there. In case of conventional, you can say you are having non-specific binding also, primer dimer formation also, but in case of hot start PCR, you don't have other impurities. You are having only product, whatever product you want, whatever product you desire. So to restrict the non-specific binding, we are doing this one only. There are different way by which we can avoid non-specific binding. I have told you four stages are there, four different things are there by which you can restrict it. To do those four processes, you can do either inactivating the or inhibition of the enzymes that we have seen just now by using the wax, or you can later on add the DNA polymerase. DNA polymerase I will not add in the first step. I will add DNA polymerase after the second stage onwards. Yeah, after heating process I will add it. So when you want to inactivate, you can use some kind of antibody, you can use some kind of oligonucleotide, or you can use wax. Just now we have seen. So these three way by which we can inactivate or inhibit the tag DNA polymerase. Or late addition of tag polymerase can be done by preheating, freezing of the reaction, or late addition of tag polymerase. Uh, by using enzyme linked antibody, so we are going to have the enzymes. Uh, so enzymes is associated with the antibody. Let when you are going to heat the reactions, because antibodies are the composed of primer, so this primer protein will be denatured and enzyme will be free. So after the heating reaction, enzymes becomes free and reaction will take place. So when you are going to use enzyme associated antibody, so heating will denature this antibody, enzymes becomes free and reaction will take place. Preheating of the PCR machine. Uh, In this method, heat the PCR machine at 95 degrees centigrade, prepare all the reaction on the ice at 4 degrees centigrade, and immediately put the tube into the PCR machine. This will decrease the chances of tag activation. Also, the mispaired primer or release immediately. So these are the different way by which we are doing the, you can call it a hot start PCR, different by using wax beads or high specific oligonucleotides. Uh, there are certain advantages. Hello. One of the major advantages is it decreases the non-specific binding or non-specific elongation of the DNA can be done by this hot start PCR. It also prevents mispriming or misprimer dimer formation. These are the two major advantages of this hot start PCR is there. Okay. So once you will have these two things, obviously your PCR efficiency will increase and yield will increase. There are certain disadvantages also associated with this one. The overall cost will increase because you are going to use antibody or different weeds, different kind of other assembly you are going to use it. So obviously cost will increase. Another disadvantage is it should not be nested PCR, it should be hot start PCR here. Another disadvantage is that it can amplify the larger DNA template. So hot start DNA tag DNA polymerase can be either enzyme linked you are going to use, oligonucleotide linked you can use it, antibody associated you can use it, or chemically modified enzymes you can use it. It facilitates specificity and binding non-specific. Okay. Now the last important modification is nested PCR. Nested PCR is again, this is also for the increasing the specificity only. In this case, what we are going to do, we are going to use two primer set. Means normally in the conventional, we are going to use only one primer set. In the case of nested PCR, we are going to use for the same DNA. Like if I have this DNA, I will use two different sets of primers. Like this is my DNA and this portion of DNA is of R choice. I want to elongate this portion of DNA. So what I will do, I will make two sets of primer. One primer set will bind outside the target DNA. This is my target DNA, so one will bind outside the target DNA in the first step, and the second set of DNA will be for the inside the target DNA. So remember, nested PCR we are going to use. This is the purpose is to increase the specificity. For this one, we are going to use two sets of primer. One set of primer binds non-specifically outside the target DNA, and second set of primer is going to bind with the target DNA or within the target DNA. Fine. Two sets of primer are used to achieve high specificity, high sensitivity in the nested PCR. Both primers have different and unique properties. The first set of primer binds outside the target DNA, amplifies larger fragment. Another set of primer binds specific to the target DNA, and this increases only internal primers. So this is the modification of the normal PCR where two sets of primer will be used. Like this is my target DNA. So first primer set will bind outside. This is my target DNA. So first set of primer binds outside, increases your DNA, and then second set of primer, whatever product you will get after this reaction, second set of primer will bind with that one only. Like here you can see it. This is my target DNA. In this case, I want this DNA to be amplified. So first set of primer, this is my outer primer, which will bind outside the target DNA here and here. This is going to elongate some DNA, some pieces of DNA. Whatever product you will get after the first primer, then we are going to use second set of primer for the binding. So second set of primer we will get after first. This techniques, this nested PCR does not require any additional reagent or chemical or instrumentation besides conventional. So whatever product you require whatever component you require during the conventional PCR, same component we are going to use here also. Only one thing you require extra, that is one extra set of primer. One in conventional we are using only one set of primer. Here we are using two sets of primer. That is the only difference. Remaining everything are same. The outer set of primers are the primer that is binding to the upstream to the inner set of primer. So this is inner set of primer. So before that one binds outer primer. Then we are having inner set of primers. The inner set of primer is non-specific. It will restrict the non-specific binding and will not amplify the second set of amplicon. Second set of primer is specific to the inner sequence or the target sequence you can call it, and this will only elongate your target DNA. Like you can see here, because this is the outer primer, so this will elongate only this portion of the DNA, and this is the outer primer, so this will elongate only this set of primer. So this portion of DNA has been left out, so this is not amplified by first primer. In the second set of, when you have got only this portion will be amplified by the in the first step. This will use for second round of PCR by second primer. So this is in the case of this one, second set of primer will bind here and elongate only this portion of DNA. So rather than elongate a complete set of DNA multiple times, we are going to restrict elongation of only the specific region of the primer. So only so specificity will be increased by using two sets of primer. Uh, Here, this is the step. Different scientists are given different steps. So here, if you'll say PCR steps are the temperature. For initial denaturation, we are using 90 to 95 degrees centigrade. Denaturation for then annealing, then extension, and then final extension. So roughly after 25 cycle, we are running for first PCR cycle with the first primer, and then 35 cycle we are running with the second primer set. What is the advantages? It is beneficial in studies some of the phylogenetic analysis and genetic polymorphism. It increases, gives 100% accuracy, having the sensitivity and specificity. So two primer different PCR like hot start and this one gives you a specificity. The impossible templates where the GC content might be high or chances of the non-specific binding is higher, nested PCR offers the best result. It is useful for the amplification of gene with the low abundance, and nested PCR is the best choice for carcinoma and viral infection studies. Okay. This method is time consuming because you have to run PCR two times, one with the first set of primer, second time with the second set of primer. So it requires double time than the normal PCR. That is the one major disadvantage of this one is. Second, it is also requiring reagent two times because you have to run two times. You have to run PCR two times, gel electrophoresis two times, and because you are using DNA two times, so their chances of contamination is also a little bit higher. This PCR, nested PCR, has been used tremendously for the microbial identifications, where universal primer we are using for 16S ribosomal RNA and 18S ribosomal RNA, because these are normally conserved ribosomal RNA. So we are going to use this 16S ribosomal RNA, 16, 18S ribosomal RNA for the study of microbial identification and phylogenetic analysis. Like this is how it will be done. We have four different species. We are running 16S ribosomal RNA gene for four different organisms. Reaction preparation from the universal primers. Means for this one we are going to have two pair of primer. For this one also two pair of primer. After first round of universal primer we are getting this one. Second round we are getting this one, and then we are going to, by gel electrophoresis we are going to identify. Like here you can see. Okay. Now, so these are the three major variants of the PCR. One was multiplex, second was hot start, and third one was nested. There are some other variants like two-tailed PCR, where you can see where you are going to use one hemi probe, uh, which is because same primer we are going to use only one means primer, whose both side strands will be used as a primer. So this is going to bind at one place and this is going to bind at the another place. So the only one set of primer, only one primer will work binds at two different places. So that's why it is called as two-tailed PCR. Yeah, two-tailed. You are using single primer which binds at the microRNA target of both 3' and 5', known as hemiprobes. So that is, then we are for bacterial colony identification, we are doing colony PCR, where we are multiple colonies can be identified. Then we are using reverse transcripted PCR, RT-PCR. This we have used too much in during this COVID times, for the where RNA will be converted into cDNA by using reverse transcriptase, and that cDNA will be used for PCR. So first messenger RNA we are taking. Means we have two types of RT-PCR. One is one step, second one is two step. In one step, we are taking messenger RNA, converting by reverse transcriptase into DNA, and then DNA will be used for PCR cycle, PCR reaction. So in the single tube, at the first we are going to use all the components which is required for PCR plus reverse transcriptase. So no need of pipetting second time. Where in case of two set PCR, first process we are doing the reverse transcriptase. This process will be reverse transcription. After this reverse transcription, we are taking RNA, the cDNA, and then using the PCR reaction. So reverse transcription separately done, and PCR process will be done separately. So that's why it is called as two step. One step for reverse transcription, second step for the PCR. In this case, we don't have two separate steps. This is a single step. So process will be the same. Only thing is we are doing two process separately, or two process together. Then we are having asymmetric PCR, where primer means primer length will be one primer we are going to use in abundant quantity, and second primer will be in the very limited number of primers. Like you can see here, one primer we are going to use it four times higher than the second primer. So because this primer we are going to use in too much higher quantity, so obviously the elongation of this DNA will be too much. So this is called as asymmetric elongation of the DNA fragment. Then we are having touchdown primers, where every step we are going to annealing temperature is gradually decreases. Like in this case, you can see annealing temperature is like x degree, so that is the melting temperature plus 10 degree. In the second cycle, the annealing temperature has been reduced here. Third cycle, and again annealing temperature has been reduced to Tm. So gradually, as a cycle is going on, annealing temperature has been decreased. So reduction in the annealing temperature. This is called as touchdown PCR. Then ligation mediated PCR, where we are going to use linkers, methylation specific PCR, where DNA methylation of cytosine and guanine can be detected, cold PCR, then assembly PCR. So these are different variants, and this is not only this one. There are many number of variants are there according to the requirement. Whenever we require one kind of thing, we modify the PCR. So these are detailed about the PCR, their variants. So if you have any questions, you can ask. I will slides I will share with you. Any question anyone is having? No question? No one is having any question, right? Fine. Anyway, whenever you are having question, you can message me or you can type me. I will give it to you.